Digestion is important for breaking down food into nutrients, which the body uses for energy, growth and cell repair. The body breaks down nutrients from food and drink into carbohydrates, proteins, fat, minerals and vitamins. Carbohydrates. Carbohydrates are sugars, starches and fibers found in many foods. They may be simple or complex depending on the chemical structure. Simple carbohydrates include sugars found naturally in foods such as fruits, vegetables, while complex carbohydrates are starches and fiber found in whole grain breads and cereals, starchy vegetables and legumes. The main function of carbohydrates is to provide the body with energy to support the muscular work, brain activity, breathing and other important activities. Carbohydrates are made up of sugars known as saccharides. Most carbohydrate foods contain many saccharides linked together which are known as polysaccharides. Carbohydrate digestion begins in the mouth and is complete when the carbohydrates are broken down into single sugars or monosaccharides, which can be absorbed by the body. Proteins are important nutrients that the body uses to build cellular structures that carry out vital functions. Foods such as meat, eggs and beans consists of large molecules of protein that the body digests into smaller molecules called amino acids. The body absorbs amino acids through the small intestine into the hepatic portal system, which then carries them throughout the body. Lipid is defined as a fat-like molecule that does not have the ability to dissolve in water and hence tends to clump together and form large droplets as it moves to the digestive system. During digestion, the body breaks down fat molecules into fatty acids and glycerol. Fat molecules are a rich source of energy for the body and help the body absorb vitamins. Oils such as corn, canola, olive, soybean and sunflower are examples of healthy fats. The mechanical and chemical digestion of food takes place in many steps, beginning in the mouth and ending in the large intestine. Digestion of carbohydrates is performed by several enzymes and begins in the mouth. The salivary gland in the mouth secretes saliva, which helps to moisten the food and release the salivary amylase or ptyalin enzyme. It is a mixture of two enzymes, alpha and beta amylase, which hydrolyze polysaccharides into erythrodextrin, archodextrin and finally into maltose in presence of chloride ion. The optimum pH is 5.5 to 6.5. Insignificant amount of maltase is present which converts maltose into glucose. The food is now called bolus. The bolus is swallowed and passed through the esophagus. The mixture enters the stomach where it is known as chyme. There is no further digestion of carbohydrates in chyme, as the stomach produces acid which destroys bacteria in the food and stops the action of salivary amylase. The next step of carbohydrate digestion takes place in the duodenum. In response to chyme being in the duodenum, the pancreas releases the enzyme pancreatic amylase that hydrolyzes starch and glycogen to alpha-dextrin, which are then digested by glucoamylase into maltose and maltotriose. The small intestine then produces amylolytic enzymes in succus entericus called lactase, sucrase and maltase. Sucrase hydrolyzes sucrose into glucose and fructose. Maltase and isomaltase hydrolyze maltose and isomaltose respectively to glucose and lactase. Hydrolyzes lactose to glucose and galactose. Large intestine. Carbohydrates that were not digested and absorbed by the small intestine, including resistant starch foods that reach the colon, where they are partly broken down by intestinal bacteria. Fiber, which cannot be digested like other carbohydrates, is excreted with feces or partly digested by intestinal bacteria. Cellulose and certain other plant complex carbohydrates cannot be digested by humans due to lack of enzymes to hydrolyze them. Certain fiber types help to speed the movement of stool through the colon. Protein digestion is a multi-step process that begins in the mouth and continues through the intestine. Proteins are digested into amino acids and small peptide, dipeptides or tripeptides by hydrolyzing the peptide bonds. Stomach. A large part of protein digestion takes place in the stomach. There are two groups of peptidases, endopeptidases, which attack the internal peptide bonds and release peptide fragments, for example, pepsin and trypsin. Exopeptidases, which act on the peptide bonds of terminal amino acids, for example, carboxypeptidases. The gastric juice in stomach contains pepsinogen enzyme which is activated to pepsin by HCl with an optimal pH of 2. Pepsin breaks the peptide bond between tyrosine and phenylalanine amino acids, thereby producing peptide fragments having short chains of 4 to 9 amino acids and called peptones. Proteins are converted to acid metaprotein, which gets converted to primary proteoses, then secondary proteoses, and finally into peptones. The gastric mucosa in infants secrete enzyme called prorenin, which is converted to renin by HCl, which breaks down milk proteins or casein to soluble paracasein and then to insoluble calcium paracaseinate in presence of calcium. Casein and renin in presence of HCl is converted to paracasein, which in presence of calcium ion converts to calcium paracaseinate. HCl helps to kill bacteria in food that could cause infection. It also makes the stomach very acidic with a pH of 1.5 necessary to activate the enzymes. Small intestine. As the chyme meets the neutral pH of the duodenum, pepsin is denatured, rendering it inactive. The pancreatic proteolytic enzyme of pancreatic juice called trypsinogen is activated by enterokinase located in the membrane along the lining of duodenum. It converts trypsinogen into its active form trypsin that catalyzes the conversion of both chymotrypsinogen and procarboxypeptidase into their active forms, chymotrypsin and carboxypeptidase respectively. Trypsin acts on the peptides and polypeptides, reducing them to smaller peptides by hydrolysis. Chymotrypsin hydrolyzes peptide linkages with carboxyl group of aromatic amino acids like tryptophan, tyrosine and phenylalanine, finally producing dipeptides and tripeptides. Carboxypeptidase specifically hydrolyzes the amino acids from the carboxyl end of peptide chains to yield individual amino acids. Proteins are converted to alkali metaproteins, which are then converted to primary proteoses and secondary proteoses. These then finally convert to peptones, then polypeptides and into amino acids. Proelastase, protaminase, collagenase present in pancreatic juice when activated break down elastin, protamines and collagen into simpler forms. The small peptides are transported into the cell lining, the small intestine, where intracellular peptidases, that is the brush border enzymes called aminopeptidase and dipeptidase, continue the digestion of peptides and convert them into amino acids that finally enter the blood. The proteolytic enzymes present in the intestinal juice are enterokinase, aminopeptidase, prolidase, tri and dipeptidases. Enterokinase activates trypsin in the presence of calcium as cited earlier. Aminopeptidases are capable of removing one amino acid from N-terminal end of the peptide. They cannot hydrolyze the linkages of dipeptides or if the N-terminal amino acid is proline. Prolidases are enzymes that hydrolyzes the linkages involving proline. Thus, by the concerted action of the enzymes, proteins are broken down to di and tripeptides. Di and tripeptidases present in the intestinal mucosal cells or inside the absorptive cells cleave them to amino acids. Most of the proteins are digested in the duodenum and jejunum by the proteolytic enzymes of pancreatic juice and succus entericus. Lipid digestion begins in the mouth, continues in the stomach and ends in the small intestine. Most dietary fat is in the form of neutral fats or triglycerides. Each triglyceride molecule is broken down into one monoglyceride or acylglycerols and two free fatty acids by lipase enzymes. Most lipid digestion occurs in the duodenum. There are several lipases such as lingual lipase, gastric lipase, breast milk lipase, pancreatic lipase, cholesterol esterase and phospholipases A1 and A2. In mouth, the lipid digestion in mouth is performed by lingual lipase, produced and secreted by serous lingual glands, also called von Ebner's glands. Lingual lipase is best suited to act on milk. It plays a modest role in the digestion of triacylglycerols. Lingual lipase activity is not particularly important for healthy adults, rather it is important for infants, where pancreatic lipase is still immature. The enzymes can account for 10 to 30% of triacylglycerol hydrolysis occurring in the gastrointestinal tract and up to 50% in breastfed infants. Stomach. The lingual lipase is also stable in an acid environment and therefore remains active in stomach. It acts on the food in the small intestine in case there is no proper pancreatic secretion of bicarbonate. It breaks down lipid into single fatty acid and 1,2-diacylglycerol, which is then hydrolyzed in the duodenum. Gastric lipase present in the gastric juice is very weak and is activated by calcium ion. It degrades fat containing short chain fatty acid at neutral pH. The digestion of lipid in the stomach of an adult is almost negligible, since lipids are not emulsified and made ready for lipase action. The lipids end to coalesce in stomach, forming a separate phase from the surrounding environment, thus limiting the opportunities for the enzyme to hydrolyze triacylglycerols into short chain and medium chain fatty acids and diacylglycerols amphipathic molecules. Due to the action of surfactants together with the churning action of the stomach, emulsion of fat droplets are obtained with a hydrophilic surface. This leads to decrease in size and then released into the duodenum as chyme. In small intestine. In small intestine, in the duodenum, the hydrolysis of triglycerides continues. The chyme is mixed with bile that emulsifies lipid droplets, increasing the surface area for hydrolase activity. Cholecystokinin stimulates the exocrine pancreas to secrete pancreatic juice containing pancreatic lipase. The enzyme catalyzes the digestion of the majority of ingested triglycerides, mainly in the upper portion of the jejunum, and has a pH optimum of 7.0 to 8.8. Its activity is increased in the presence of potassium ion and bile salts. It catalyzes the cleavage of fatty acids, typically with 10 or more carbon atoms. Lipase digests triglycerides into monoglycerides or 2 monoacylglycerols and free fatty acids. The digestive enzyme lipase is water soluble and can only work at the surface of fat globules. Motility in the small intestine breaks fat globules apart into small droplets, that are coated with bile salts and phospholipids, preventing the emulsion droplets from reassociating. Other amphipathic molecules present in food are lecithin and phospholipids, and all together increase the surface area for hydrolase activity. Another factor that helps is co-lipase, an amphipathic protein that binds and anchors lipase at the surface of the emulsion droplet. Another enzyme present in pancreatic juice is cholesterol esterase. This enzyme acts on cholesterol esters and releases cholesterol and free fatty acids. The digestion of phospholipids is carried out by phospholipases, primarily phospholipase A2. This enzyme present in the pancreatic juice in the form of zymogen and is activated by trypsin in the intestinal lumen. It catalyzes the cleavage of fatty acid of phospholipids. Phospholipase A1 removes the fatty acid of the phospholipid. In the intestinal mucosa, the enzyme phospholipase B or retinyl ester hydrolase is active on vitamin A esters. The digestion of phospholipids end with the formation of a free fatty acid and lysophospholipid. The absorption of most digested food occurs in the small intestine through the brush border of the epithelium covering the villi and microvilli. The mucosa also contains special cells that increase the absorption of nutrients. It is not a simple diffusion of substances, but is active and requires energy used by the epithelial cells. All carbohydrates are absorbed in the small intestine and into the bloodstream in their monosaccharide form, such as glucose, galactose, xylose, fructose, and mannose, by passive and active diffusion. The absorption of carbohydrates is usually in the upper small intestine by the portal system. However, when the diet contains carbohydrates not easily digestible, digestion and absorption takes place mainly in the ileal portion of the intestine. Fructose is absorbed and transported into the intestinal cells cytosol. Glucose and galactose competes with other sodium transporter required for absorption. From the cytosol, monosaccharides pass into capillaries by simple or facilitated diffusion. The bacterial flora metabolize these compounds anaerobically in the absence of oxygen. This produces gases such as hydrogen, carbon dioxide and methane, and short-chain fatty acids such as acetate, propionate and butyrate. Fatty acids are rapidly metabolized. Butyrate, used mainly by the cells in the colon, and acetate is absorbed into the blood and taken up by the liver, muscle and other tissues. Propionate is an important precursor of glucose in some animals, but not humans. Any remaining sugars are acted upon by brush border enzymes found on the microvilli of the small intestine and complete the digestion. The absorption rate of monosaccharides is in the following order: galactose, glucose, fructose, mannose, xylose and arabinose. The body uses the calories from proteins which are released during the digestion process for energy when carbohydrates and fats are not available. Proteins are absorbed in the small intestine, in the jejunum and ileum as single amino acids, dipeptides and tripeptides. A number of amino acids are co-transported into the luminal side of the epithelial cells with sodium via secondary active transport. There are a number of different amino acid transporters that requires energy. Each amino acid group has a carrier protein that is responsible for transporting it from the intestines to the mucosa cells. Sodium and potassium are minerals needed for amino acids to pass from the intestines through the villi and into the bloodstream. Most proteins is absorbed as dipeptides and tripeptides, which are further digested to single amino acids inside the GI epithelial cells. Amino acids and small peptides that are absorbed reach liver through the portal circulation. Naturally occurring L amino acids are absorbed actively, while D amino acids are absorbed passively. Absorption of amino acids is similar to those of carbohydrates, and they need a carrier and sodium ions. Amino acids are absorbed via glutathione cycle. Very small amount of amino acids may enter the lymphatics. Lipases break down lipids into fatty acids and glycerides. These molecules can pass through the plasma membrane of the cell, entering the epithelial cells of the intestinal lining. The bile salts surround long-chain fatty acids and monoglycerides, forming tiny spheres called micelles. The micelles move into the brush border of small intestine absorptive cells, where the long-chain fatty acids and monoglycerides diffuse out into the absorptive cells, leaving the micelles behind in the chyme. The long-chain fatty acids and monoglycerides recombine in the absorptive cells to form triglycerides, which aggregate into globules and are then coated with proteins. These large spheres are called chylomicrons. Chylomicrons contain triglycerides, cholesterol and other lipids that have proteins on their surface. The surface is also composed of hydrophilic phosphate heads of phospholipid. Together, they enable the chylomicron to move in an aqueous environment without exposing the lipids to water. As chylomicrons are larger in size, they cannot pass through blood capillaries, so these lipid materials enter the lymph vessels or lacteals and then transferred into blood from lip. Chylomicrons are released by exocytosis at the basolateral surface of the enterocytes or absorptive cells and flow into circulation via lymphatic vessels, which drain into the general circulation through the portal vein. The breakdown products of lipids are absorbed by simple or passive diffusion through the phospholipids bilayer of the intestinal epithelial cells. Most polar substances are absorbed using either active or passive carrier-mediated transport mechanisms. The colon absorbs small quantities of salt and water as the feces are concentrated there and eventually eliminated through the rectum and anus. Absorption takes place through the mucosal lining of the small intestine and enters the epithelial cells. Once inside the cells, the free fatty acids and monoglycerides enter the smooth endoplasmic reticulum. Here, the digested products are resynthesized into triglycerides, the major form of stored fat. The absorption disorders, also called malabsorption syndromes, are characterized by problems digesting or absorbing substance in the diet. There are a number of different types of absorption disorders, most of which result in a decreased ability to absorb nutrients. Disorders that increase the absorption of certain nutrients include hemochromatosis or iron overload and Wilson's disease, where there is increased copper absorption. Common absorption disorders include celiac disease, which is caused by intolerance to gluten, and lactose intolerance. Lactose intolerance is an inability to break down sugar in dairy products, such as lactose. It is caused by a lack of enzyme called lactase that helps convert lactose into a form that can be used by the body, such as dextrose and galactose. Lactose intolerance is also called primary or secondary lactase deficiency. Primary lactase deficiency is a common condition that develops over time as the body produces less lactase. This process begins around 2 years of age, but symptoms usually develop much later in life. Secondary lactase deficiency occurs when damage to the small intestine reduces the production of lactase. Conditions that can cause secondary lactase deficiency include celiac disease and inflammatory bowel disease, for example, Crohn's disease and ulcerative colitis. Other types of absorption disorders include tropical sprue and Whipple's disease. Tropical sprue is more common in tropical and subtropical areas of the world, including the Caribbean and Southeast Asia. The exact cause for the condition is unknown, but it may be related to an infection that damages the lining of the small intestine. The symptoms include anemia, diarrhea, weight loss and malnutrition. It is treated with antibiotics. Whipple's disease is caused by bacteria called Triforma whippleyi that usually infect the small intestine and cause malabsorption. Whipple's disease also can affect the areas of the body like joints, eyes, heart, lungs and brain and cause serious complications. Patients who have Whipple's disease often experience joint pain and stiffness years before developing intestinal symptoms such as severe diarrhea, GI bleeding and abdominal pain. Treatment involves antibiotics and may also involve intravenous fluid and electrolyte replacement along with dietary supplements. If left untreated, Whipple's disease can be fatal. Many conditions can cause nutrient malabsorption. Celiac disease, gluten sensitivity, lactase deficiency, infection, parasites, chronic diarrhea and others. If our body doesn't absorb nutrients well, we could experience nutrient deficiencies or improper absorption, taking in too much or too little of certain nutrients. The possible reasons for poor nutrient absorption include damaged mucosal barrier, absence of certain enzymes, poor circulation of bile or related acids, defective detoxification or iron transport or pancreatic insufficiency, anemia, gallstones or kidney stones, osteoporosis, malnutrition or weakened immune system. Inadequate nutrient absorption can contribute to other systemic problems such as inflammatory joint disease, chronic dermatological disorders, chronic inflammation and sensitivities. The diagnostic tests include hydrogen breath test, stool composition, enzyme production and some advanced imaging techniques. Carbohydrates, fats and proteins are the major nutrients in food that cannot be utilized by our body in their original form. They have to be broken down and converted into simple substances in the digestive system, both mechanically and chemically. In the mouth, carbohydrates are broken down by amylase into maltose. In the duodenum, disaccharides are broken down into monosaccharides by enzymes and then absorbed into the bloodstream and transported to the cells to be used in metabolic pathways to harness energy. In the stomach, proteins are broken down to peptides, which are then broken down into single amino acids and absorbed in the small intestine. Lipids are digested mainly in the small intestine by bile salts through the process of emulsification and lipases into fatty acids and monoglycerides. These are then absorbed in the small intestine through miceles and recombine into chylomicrons in the bloodstream.